Welcome to video 4 of topic 3 IB Biology Genetics. In this video we are going to be looking at 3.4 inheritance. As you can see here from our study list, you'll see that 3 areas are already ticked off. That's because we have studied those in previous units. So although they'll be covered in this, they won't be directly covered. Okay, so we'll start off looking at our understandings to build our knowledge ready for the skills and application. Before we get to that, there's many key terms in this unit that you need to be aware of that you already would have heard in previous units. So we'll be talking about genes, chromosomes, DNA, proteins, nuclei, cell. So all that kind of terminology based around our inheritance, okay? From previous years, you would know what chromatids are, the centromeres at the center, knowing that the chromosomes replicate. We get then those homologous chromosomes. We have sister chromatids through the replication, okay? Other terms that you should be aware of is this whole concept of the alleles. Okay, so when you've got your two homologous chromosomes, you have genes, the same gene on each chromosome, and because they may be a slightly different form, they're called alleles. Just remembering that the location of the gene or allele on the chromosome is called the locus or loci. Some more terminology that you should be aware of. Remembering what I was saying, this is all about these key terms, this unit, and really getting to understand those. We have homozygous chromosomes, where we have two genes that are the same. So for example here, or the alleles are the same, sorry. So we've got two dominant alleles on that first example, the two capital Bs. We have our heterozygous chromosomes or alleles, where we've got our capital B, lowercase b, so dominant and recessive. And then we have our homozygous, where we have two recessive alleles. Just remembering those terminologies, homozygous, heterozygous, homo meaning the same, hetero different. Remembering that our gametes, our sperm cell and egg cell, have half the chromosome number of the zygote. We've got human sperm cell, egg cell here with the 23 chromosomes, which on fertilization gives us the 46. Okay, now part of your course is your understanding of the nature of science. And for this section in 3.4, that is making quantitative measurements with replicates to ensure reliability. So the more we repeat an experiment, the more reliable it will be. So when you're looking at IAs, you need to ensure that if you're doing a practical IA, you carry out a suitable number of repetitions to ensure reliability of your results. Now Mendel, when he was looking at pea plants, he carried out many replications, okay, with his genetic crosses to give him a considerable amount of numerical data. So rather than just using qualitative analysis where he just looked at the characteristics, Gregor Mendel also collected large amounts of quantitative data, so he then had that numerical evidence. This allowed him to reach then valid conclusions. Your information needs to be reliable to reach those valid conclusions. Now Mendel discovered the principles of inheritance with experiments in which large numbers of pea plants were crossed. So peas have some very clear characteristics. One of those is flower colour. Okay, and this can easily be followed from one generation to the next. They can then be crossed to produce hybrids, and they can be allowed to self-pollinate. So it gives the opportunity for lots and lots of different genetic outcomes and genetic traits to be carried on. Now, Mendel was a pioneer in obtaining quantitative results and in having large numbers of replicas. Okay, so there were 7 different crosses that he could use, 7 different investigations with his pea plants. Okay, so he could look at the seed, where he's looking at the form of the seed, whether it was rounded, whether it was root-cooled. He could look at the cotyledon, which is like the very first sprout that comes out of the seeds, whether it has 1 or 2 leaves. Here, pea plants are what we call dicotyledon. They have 2 little leaves, but whether those little leaves are yellow or green. The flower colour, so we've got white, we've got violet, and then we can have the cross. We've got the pod, okay, so the form, whether it's a nice full pod or whether it's constricted, the colour of the pod. He could look at the stems, so the place of where the axil pods were, the terminal pods were, the size, whether it was tall or whether it's short. So there's all these different features or characteristics that he could test through his work with genetics. So it gave him a whole extensive range of data. Okay, so when we're thinking of this whole concept of alleles, and we've talked that we've got dominant, we've talked that we've got recessive. We've got our pea plants here. Okay, and we can see we've got dominant and recessive genes. All right, so dominant alleles can mask the effects of recessive alleles, but codominant alleles have joint effects. So basically, what that's saying is if we've got dominant alleles, then the dominant feature will be there. If we've got recessive alleles, we can get the recessive trait, but then we can get this codominance as well. Now, the usual reason for dominance of one allele is that the allele codes for a protein that is active and carries out a specific function, whereas the recessive allele codes for a non-functional protein. So unless you've got 2 recessive, then it won't be functional. If you've got 2 recessive, then one will become functional. If an individual heterozygous, so for example, capital B, lowercase b, so we've got our dominant and our recessive, heterozygous different, then it will be the allele for the dominant trait that is displayed, masking the effect of the allele for the recessive trait. So the dominant trait will always be the strongest as such. Only one copy of a dominant allele is required to display that trait. So it's going to be your 2 capitals or your capital and your lowercase, okay? Don't say, like, I know I just slipped up and I said it is the strongest. You're talking functional proteins now, okay? For recessive traits to be displayed, you need 2 copies of that recessive trait, okay? So you need 2 recessive alleles. Okay, so what is it about codominance? And you can see here we've got some flowers, we've got the pink and the white. Well, the codominant shows both those colours coming through. So codominant genes have pairs of alleles where both have an effect when present together. So this is our example, our flower, the Mirabilis jalapa. So in this flower, we've got our allele for red, which is CR. And we've got, so the C could stand for colour, whatever. We've got our allele for white, which is CW. So if we get if we get double CRs, we get a red flower. If we get double CWs, we get white flowers. If we get a CR and a CW, we get a pink flower. Okay, now you'll see that both of the R and the W are both capital. Okay, they're both dominant. So when you get the 2 together, that's why we get codominance. Alrighty, so we've had a think about these alleles and the alleles being different, and in a previous unit we talked about the difference in the allele was actually due to some kind of mutation at some point, a change in that base sequence. We know that in humans we do get changes in the base sequence, so differences in genes, and in some cases this can cause our genetic diseases. Now, many genetic diseases have been identified in humans, but most are very rare, as they are caused by a very rare recessive allele. The chance of inheriting one allele for any specific disease is small, but to develop the disease in most cases you need 2 alleles. So if they're both inherited, it shows how that's that extremely small chance of getting both of those alleles from both parents. Remember, we're talking recessive here. There are examples. We're going to go into all these examples, and there are some that are dominant diseases and things like that, but we're just talking basically at the moment. The diagram on the right, don't take too much note to that. It's just showing you a range of genetic diseases linked to each human chromosome. Now, changes in genetic sequences can lead to genetic diseases in humans, and we call those mutations. So we spoke about this in a previous unit. Okay, now many genetic diseases in humans are due to recessive alleles of autosomal genes. Now, the autosomal genes are our normal genes. Remembering we've got, our normal chromosomes are autosomal, and then we've got our sex chromosomes. Okay, so we're talking autosomal. So we are talking the chromosomes that are not the sex chromosomes. We're talking about genes that are on there that are not sex chromosomes. Okay, so although some genetic diseases are due to dominant and codominant alleles, so we'll get to them in a minute. Let's have a look at our ideas around recessive and dominant here. So we're on autosomal. Now, if we've got autosomal dominant, which you'll see on the left-hand side here, you've got one dominant gene there in one affected parent. Okay, now with the offspring, if they've got that dominant gene, they will be affected, right? They don't need two of them, they just need one of them because it's dominant. They will be affected. If they don't have any dominant genes, they're not affected. Okay, so it's as simple as that with autosomal dominance. They've either got the gene or they haven't. They've either got the condition or they haven't. Now, it's with recessive that it gets a bit trickier, okay, because with recessive, this is where we can have carriers. Now, the gene is in blue, okay, and it's recessive this time, remember? Now, to actually be affected, you need two recessive affected genes. So that's the offspring on the right hand side. However, the two offspring in the middle there, you'll see they've got the gene, but because it's recessive, they just carry it. And then the child on the left hand side doesn't have the gene, so it's not affected. Okay, now looking at codominance, this is giving us an example of some blood types here, which we will look at later on. Okay, you can see that we've got an allele for the A blood type, an allele for the B blood type, and an allele for the O blood type. Now, A and B are dominant. Okay, so if we look at our offspring, on the far left, we've got offspring with an A allele and an O allele. A is dominant, blood type A. Next, we've got offspring with an A and a B. Now, they're both dominant, so we have a codominant, an AB blood type. The third child, or second from the right, they have got a B and an O, so they're B because B is dominant over O. And then on the right hand side there, we've got two O alleles, so they've got blood type O. Okay, so having a closer look at autosomal recessive diseases, and two examples we have there is our cystic fibrosis and our sickle cell anemia. Now, we spoke about sickle cell anemia in a previous video, okay, and then we've got cystic fibrosis. So both are carried on recessive alleles, okay? So with both of these diseases, you can have carriers. You need two recessive alleles of the condition to actually be affected by the condition. So recessive, you can have carriers. Two examples of cystic fibrosis and sickle cell anemia. All right, now some dominant autosomal diseases. Okay, so our example here is our Huntington's disease. Now, with our dominant, you've either got it or you haven't. Okay, so you can see the offspring here. We've got a 50-50 chance of the offspring being affected or not being affected. So autosomal dominant, and we're looking at Huntington's disease. You've also got the pedigree there. On the right hand side, we'll be talking more about pedigrees later in the video. Now, all those conditions we've spoken about so far are autosomal linked. Now let's have a think about sex linked. Okay, so here we've got two images of X-linked dominant. Okay, so we're talking the X chromosomes, remembering that the females have double X and the male has XY. Okay, so if the female is carrying the dominant gene, so the female is infected. If you now look at the offspring, now any sons will inherit the Y chromosome from the father. Okay, they can still inherit that X chromosome, which is infected, has the infected allele on, from the mother. Okay, so there is still a chance that they could be affected. All right, and so there's both males and females can end up affected because we are talking X dominant. Now, if the father has the affected allele on the X chromosome, it's 100% guaranteed that any daughters will be affected because they inherit that X chromosome. Okay, whereas the sons on the, if the mother's got the dominant gene, the affected gene, then the sons could inherit either of those X chromosomes. Okay, so daughters from an affected father with an X-linked dominant condition will be affected. Okay, so X-linked recessive. So we'll look at the mother being the carrier first up. So we've got carrier mother and an unaffected father. Okay, so what could occur here? We could end up with a carrier daughter. We could end up with an affected son because there is no sorry, lost the words here. They will only have the affected allele on one X chromosome. So if they've got that on their X chromosome, then they will be affected. A daughter could end up a carrier, but a son can't. Sons have either got it or they've not. Daughters can be carriers, but they wouldn't have the condition because they'll always have the second X chromosome. Okay. If we look at an infected father, well, in an infected father, you will always end up with carrier daughters because they would have to inherit the X chromosome, but sons would always be unaffected because they get the Y chromosome from the dad. Okay, so if a condition is Y-linked, then sons are guaranteed to be affected. It will be passed from the father. The daughters will always be unaffected because they don't have that Y chromosome. So as we said, there's many genetic diseases that have been identified in humans, but most of them are rare because they are recessive. Not all of them are recessive, remember, most of them. Diseases are caused due to a mutation, okay, and two things that can cause these mutations can be radiation and mutagenic chemicals. Although, remembering what we watched in a previous video, mutations can just occur over time as well. However, radiation and mutagenic chemicals are big cases of causing mutations. So mutation is where we have that random change in our base sequence which creates that new allele. They can be caused by radiation, where energy causes chemical changes in the DNA. So gamma rays and alpha particles from radioactive isotopes, shortwave ultraviolet radiation and X-rays are all mutagenic. Some chemicals can cause changes in DNA and are also therefore known as mutagenic, such as benzopyrene and nitrosamines found in tobacco smoke, and also mustard gas that was used in the First World War. A random change to an allele that is developed by evolution over perhaps millions of years is unlikely to be beneficial. Almost all mutations are either neutral or they can be harmful. For example, a mutation of genes that control cell division can cause a cell to easily divide endlessly, which leads to our tumours and cancer. All right, moving on then to our applications and skills. So first of all, you need to be able to know how to construct your Punnett squares and see the outcomes of monohybrid genetic crosses. So you choose your letter to represent your alleles, you determine the parents' genotypes, you determine the gametes, you draw your Punnett grid, you interpret your grid, and from there do whatever you need to do. Always choose a letter to represent the gene where it's uppercase and lowercase is easily distinguishable. Try not to use S, O, P because it's hard to tell if they're uppercase and lowercase if your handwriting's not particularly clear. Okay, your next skill, comparison of predicted and actual outcomes of genetic crosses using real data. Basically, that means you can use your Punnett squares to give you some information. So a dominant phenotype could have two possible genotypes, could be homozygous or heterozygous. A cross with the recessive phenotype will determine the phenotype of the dominant individual. This is what we call a test cross. So your skill with comparison of your predicted and actual outcomes of genetic crosses using real data. So if we have a look here, we're looking at some eye colours. If we, first up, we've got the crossing of brown and blue where we've got homozygous against homozygous, but here we've got heterozygous against homozygous. Okay, so you can see then the difference in outcomes. Because we're crossing homozygous dominant here, our outcome will be heterozygous 100%, and therefore the dominant trait will show. However, because we've got the heterozygous cross here, that alters the outcome. Okay, application, inheritance of ABO blood groups. Okay, so we spoke about this slightly earlier and we said that AB is that codominant group. So we've got 4 different blood groups. We've got A, B, AB and O. The inheritance of ABO blood groups involves both codominance and multiple alleles. Okay, if there are more than 2 alleles of a gene, they are called multiple alleles. So we're seeing that this blood group decision is not as easy as this, inheritance is not as straightforward as it looked. There are 3 alleles for ABO. Okay, we've got the A allele, which is IA, we've got the B allele, which is IB, and then we have the O allele, which is our lowercase I. Okay, so I'm just going to go back to that blood group. So if we look at the Punnett square here, we can see the different outcomes we could have. So you've got 3 possible outcomes now. So you've got your IA, so you could have IAIA, you could have IAIB, or you could have IA lowercase I as such. So we've just extended the Punnett square because we've got 3 outcomes. All right, now we are looking at colour blindness and haemophilia as examples of sex-linked inheritance. Okay, so these are both X-linked. They are both recessive. Let's look further at the colour blindness. Colour blindness is inherited. X and Y chromosomes are non-homologous. Okay, so we know that. The red-green gene is carried at XQ28. So there it is, down the bottom on the X chromosome. This locus is, so locus meaning location, is in the non-homologous region. So there is no corresponding gene or allele on the Y chromosome. Normal vision is dominant over colour blindness. Okay, so you can have the normal female or the affected female. A normal male or an affected male. It's got to have, it's on the X chromosome, okay, and it's recessive. And here we've got that carrier female. Okay, haemophilia. It's a sex-linked disease. You would have studied this in year 10. Okay, you've got your parents and you've got a father with haemophilia and the mother's not a carrier. Remembering daughters can be carriers. Okay, if you had a mother that was a carrier and a father that was a carrier, a father with haemophilia, you could potentially get daughters with haemophilia. All right, so if you look at your Punnett square of the outcome, so what do we got here? We've got our sex chromosomes, our X and our Y, knowing that haemophilia is linked here. Okay, this is a carrier, this is normal, this is normal, this has the disease, the condition. Okay, now looking at the inheritance of cystic fibrosis and Huntington's. So cystic fibrosis is autosomal recessive. We've already said that earlier in the video. Okay, which means if it's recessive, it means we can have carrier children. Huntington's is dominant, so we do not have carriers. We have affected or non-affected. We spoke about that earlier in the video. Alrighty, so one of your applications is that you can discuss the consequences of radiation after the nuclear bombing of Hiroshima and the accident of Chernobyl, and we'll look at this further in class. But just an outline, Hiroshima in 1945 was the target of the first atomic bomb to be used in warfare. And then Chernobyl in 1986, a nuclear reactor melted down, okay, and there was a massive radiation leak. So basically the things that we're looking at from this, there was a massive amount of people affected at the time, really high numbers of deaths. However, in those areas, even still today, there is extreme genetic mutation due to the radiation. We'll look at those further in class. So again, at the time, massive radiation burning, things like that. But if we're looking now, many years on, life has come back to these areas, okay? But there is a lot of genetic mutation in the species now compared to elsewhere where there hasn't been the radiation damage. Again, showing that radiation causes mutation. Again, showing how radiation can cause cancer due to the effect on DNA. This is just showing some of the different types of mutations that can occur in base sequences. Okay, so a skill now. You need to be able to look at pedigree charts and talk about patterns of inheritance. So one of the keys here is knowing all the different, knowing all the different keys on there and what they show. So homozygous, heterozygous, female, male. The key is to find the key on the pedigree that you're looking at. We'll do a lot more work around this in class. So here we've got a key. Remembering you have generations, so you start from the top where you've got generation 1 with a female and a male, and then you have their offspring from there. And each generation is listed below. So you've got generation 1, 2, 3 shown there, offspring 1, 2, 3, 4, offspring 1, 2, 3, 4, 5, 6 along the bottom. Okay, so all the little symbols there showing you different pathways that you can get on the pedigree. Okay, the symbol for our heterozygal individual can be variable. Sometimes half the square or circle is filled in. Sometimes there's a black dot in the middle. So you've just got to look at the key with the specific pedigree you're looking at. The question will have a key, okay? If you have to draw a pedigree, make sure you provide a key. You need a key to know what these things show. In questions, some information will be missing from the pedigree. You will have to deduce that information. You're going to have to work it out. 2 common questions you might see is the disorder sex-linked, or is the disorder dominant or recessive? So when giving examples from a pedigree, you must use the generation and the individual's number. So for example, you might say generation 2, individual 3. You've got generation 2, individual 4 there. So generation 2, individual 3 and generation 2, individual 4 have an affected child, which is shown in generation 3, child 2. So we've got another pedigree here. This pedigree is showing as an autosomal dominant trait, okay, because it comes up in males and females all across. There's not a pattern as such. It's dominant because, yeah, males and females are inheriting it. There's no pattern as such. Okay, what are we looking at here? We've got an autosomal recessive trait. Now, how do we know that? Well, the first lot of parents, there is no evidence of it being there. So someone there was a carrier. It affects both males and females equally. 2 unaffected parents have an affected child. All offspring of 2 affected parents must show the trait, which they do in generation 3 there. The trait may disappear and reappear in later generations. And there comes red hair. Okay, what have we got here? It is X-linked, this one, because it's present in the father first up. A male passes it on to his daughters, but none of the sons. Because remembering the father passes the Y chromosome on to the sons. Here we've got the dot as the carriers, remember. This is another X-linked, and the mother is a carrier. More males than females show this trait. All sons of the affected female show the trait. Okay, so we've worked through Unit 3.4. I will see you in 3.5.